Welcome to Standing for Truth. I am your host, Donnie, and it is a privilege to have Dr. Fuz Rana back here with me today. Dr. Rana was here approximately 2 months ago for a presentation and discussion on junk DNA. Today, we continue that discussion with a strong focus on addressing the arguments that are being used to save the junk DNA paradigm. Dr. Rana, thank you again for being here and giving us your time for this important discussion.
Well, Donnie, thanks so much for having me. I really appreciate the opportunity and look forward to the time that we're going to hang out together.
I appreciate it. I'm extremely pumped for this. This is one of my favorite topics. So definitely this is going to be exciting. We've got a great audience and we will be having questions from the audience as well, guys. So please make sure you are tagging me at Standing for Truth. We'll get to some of those questions after Dr. Rana's presentation. Now, there's also more information about Dr. Rana in the description box. I want to give him a brief introduction before we get into the presentation. Now, Dr. Rana is VP of Research and Apologetics at Reasons to Believe. Fuz earned a BS in chemistry with highest honors at West Virginia State College and a PhD in chemistry with an emphasis in biochemistry at Ohio University. You can also find a lot more about Dr. Rana, as I pointed out, including many of his must-read articles in the description box of this video. So that being said, Fuz, I think we're just going to get right into it. Unless there's something you wanted to add, we can kind of just get right into the presentation.
All right, well, it sounds good. Let me go ahead and share my screen and pull up the PowerPoint. Okay, and here we go.
There we go.
All right. And how does that look? Or is
looks
it coming?
Great.
All right, perfect.
Yeah, looks good.
Okay, so as Donnie said, I'm going to be taking a little bit of time here and just presenting some of my thoughts when it comes to the whole junk DNA phenomena and whether or not junk DNA can be rightly understood as being consistent with a case for intelligent design or a case for creation. And as Donnie mentioned a couple of months ago, I was on Standing for Truth and I presented some material there at that point in time. And so what I'm going to do is a quick reprise of some of the stuff that I presented, and then we're going to kind of get into some of the challenges that people have leveled against the case for creation, again, related to the junk DNA argument. And the first thing that I want to do is talk a little bit about why people think that junk DNA represents such a powerful case for biological evolution, for an evolutionary history of life on Earth. And there's really, in a sense, two broad reasons, from my perspective at least. The first has to do with something known as the C-value paradox. And the C-value refers to the amount of DNA that is found in the nucleus of the cells in an organism. And typically the amount of DNA is consistent from cell type to cell type in an organism, but differs from organism to organism. And you can think of the C-value as roughly equivalent to genome size. And at one point in time, people naively thought that there should be a correlation between organismal complexity and the amount of DNA in the nucleus of their cells. But lo and behold, when you plot out the C-value as a function of the different types of organisms that are found in the living realm, we see no relationship whatsoever between organismal complexity and the amount of DNA in the genome. And the resolution to this is the C-value paradox, which argues that the amount of DNA in an organism's genome has no bearing on the functionality of that DNA, but rather it is the result of an accumulation of so-called junk DNA through a variety of different mechanisms that just happen to constitute an organism's genome. So the resolution to the C-value paradox is that most of the DNA in an organism's genome is non-functional, junk DNA, and that is what explains the DNA content, not organismal complexity. And of course, when the human genome was sequenced, we discovered that the human genome harbors about 20,000 genes, which is a much smaller number than anybody would have anticipated, and that most of the human genome, somewhere between 95 to 98% of the human genome, consists of non-functional DNA. And in fact, when you look at the human genome, you can categorize the human genome uh, based on different types of so-called junk DNA sequences, and so it looks as if you can explain the structure and the composition of the human genome through, again, the C-value paradox. And then, in addition to that, the fact that we can categorize the different non-functional DNA into different classes uh, suggests to many people that we can understand this DNA again as the vestiges of an evolutionary history, where we understand mechanistically how these different types of junk DNA sequences are introduced into the human genome or the genomes of other organisms, and that we can actually organize the sequences for the different classes into these nested hierarchies, that these sequences are shared, and they also can be organized into nested hierarchies, which presumably reflect an evolutionary history. And so for many people, they see, again, junk DNA as really compelling evidence for biological evolution. It makes sense from an evolutionary paradigm, again, why the genomes of organisms contain so much junk and why that junk DNA, again, has the distribution that it has among groups of organisms that naturally cluster together. And I would actually agree that, when you look at the case for biological evolution built upon, again, junk DNA, it on the surface is a very compelling uh, case that can be made for an evolutionary history of life. But this leads me then to the next point that I want to make, and that is that one of the projects that I have at Reasons to Believe is to build a creation model for the origin and the design of genomes. I am of the conviction that there is a creator that is played a role in the origin and the basic design of life, as well as the history of life on Earth. And the reason that I have for that conviction has to do with challenges to accounting for the origin of life through chemical evolution, coupled with what I see to be an elegant design of biochemical systems. I also see elegant designs in biology. I think the evolutionary paradigm struggles to explain certain key transitions in the history of life. I won't get into those specifics. And so these are reasons why I think that an evolutionary framework is insufficient to account for the origin, the fundamental design, and even the history of life on Earth, at least in its totality. And again, I also see compelling reasons to think that there's a creator involved in the origin, design, and the history of life because of the elegant designs that I see. So the question then becomes, within that framework, is it possible to understand the human genome and the genomes of other organisms from a creation model perspective? And in order to do that, there are a couple of criteria that we have to meet. But before I get into that criteria, I just want to pause for a second and discuss a little bit about the influence of philosophy on the way in which we interpret biological phenomena. Because the framework that we see today for contemporary science, the philosophical framework, is methodological naturalism, namely that when we seek to explain phenomena in the universe and specifically phenomena within the biological sciences, we're looking for mechanistic, natural process, materialistic explanations. Any appeal to the work of a creator is excluded as a possible or a viable explanation a priori for philosophical reasons, not because that is what the evidence necessarily demands. And so within this framework of methodological naturalism, then when you look at the shared junk DNA sequences you see in genomes of organisms that naturally group together, it makes sense to think that these indeed must reflect an evolutionary history, because this is the only possible explanation that can exist. This is the only possible category of explanation that can exist. On the other hand, if we relax the requirements of methodological naturalism, we could understand that the shared features reflect common design, not common descent. And it's interesting to me that prior to Darwin's theory of evolution, the phenomena of homology was well known. And this is what we're talking about when we talk about shared biological features. These shared features are referred to as homologous structures. And Sir Richard Owen, who was a prominent biologist that preceded Darwin, was considered to be the father of comparative anatomy. He was viewed to be in the same category as an Isaac Newton in terms of his scientific prominence. And he had this idea that these shared features, these homologous structures, reflected an archetypical design that existed in the mind of the first cause that was then functionally manifested in the created order. And he conceived what he called the vertebrate archetype, that again was an idealized design that was then functionally manifested in the created order in a variety of different ways to produce a wide range of different types of biological systems and organisms. And Owen saw the archetype as being the ultimate expression of design, because he marveled at the fact that there are these archetypes that exist that are so robust that they can be varied to produce a wide range of biological systems with a wide range of function. And so this is what Owen writes: the satisfaction felt by the rightly constituted mind must ever be great in recognizing the fitness of parts for their appropriate function. But when this fitness is gained, as in the great toe of the foot of man and the ostrich, by a structure which at the same time betokens harmonious concord with a common type, the prescient operations of the one cause of all organization become strikingly manifested to our limited intelligence. So in other words, the way Owen understood homologous structures is that these reflect common design, not common descent. And what Darwin came along when he proposed this theory of evolution was he co-opted Owen's idea of the archetype, but then he evolutionized it to be a hypothetical common ancestor. And so what we see with again these shared features is that there are two different ways that they could legitimately be interpreted. Both ways actually have a precedence in the history of biology: common design or common descent. And so what we're doing when we present a creation model for the structure of genomes is that we're looking at genetic archetypes, or we would argue that the shared genetic features reflect archetypical designs that then are manifested in a wide range of different organisms, reflecting common design. Now, there's another piece of philosophical there's another piece to the philosophical influence on interpreting the biological record, and this has to do with something called the underdetermination problem. That is, it's this recognition that theories are underdetermined by data, meaning that the same set of data could equally be appropriated by two radically different types of theories and satisfy the requirements of both theories, provide support for both theories. And so I would argue that common design and common descent, given this underdetermination problem, are both equally valid ways of interpreting shared genetic features that we see in organisms, with the idea that, again, if we relax the requirements of methodological naturalism, I think the idea of common design is an equivalent theory to common descent. Now, of course, if we're going to adopt that approach, then we need to have, from a creation model perspective, a rationale for why there are junk DNA sequences in genomes, but also we have to have an ability to explain the similarities between these junk DNA sequences and the types of sequences that would be produced by a variety of mechanisms that evolutionary biologists think have generated these different types of junk DNA sequences. As an example, if we're going to argue that endogenous viral elements and endogenous retroviruses in genomes of organisms are functional, then we need to not only explain how they are functional, but also why they bear a similarity to viral genetic material. Or if we're going to say pseudogenes are functional and they are intentionally introduced into the genome as the creator's handiwork, then why do they have the appearance of, or a similar appearance to, genes? These are things that we have to do if we're going to advance, again, a creation model. Now, as we talked last time, the ENCODE project really goes a long way towards satisfying the first criteria. Namely, the ENCODE project demonstrated that a significant proportion of the human genome consists of functional DNA elements, or at least what the ENCODE project interpreted as functional DNA elements. And the ENCODE project performed a number of assays to identify functional sequences in the human genome. This includes transcription, the binding of transcription factors, histone binding, the binding of modified histones, methylation, and then three-dimensional interactions in the genome. And through this work, phase 2 of the project demonstrated that 80% of the human genome is functional, or at least displays biochemical activity that was interpreted initially as functional. And we now are at phase 4 of the ENCODE project, and phase 4 has not really produced a numerical quantitation of the percentage of the genome that's functional, but continues to affirm this original result, that a significant fraction of the human genome appears to play a functional role in genome biology. And so this, of course, is a radically different perspective on the genomes of organisms than we see when it comes to the evolutionary paradigm. And so instead of 5% of the human genome being functional, as a result of the ENCODE project, the initial result indicated 80% of it was functional, with the expectation that eventually, as we continue to do a deeper characterization, that number will go closer to 100%. Now, that's kind of a quick synopsis, a quick reprise of what we discussed the last time I was on the show. And now what we want to do is kind of get into a response that people have to the findings of the ENCODE project and the general case made by ID people and creationists with regard to, again, the functionality of junk DNA. And it's interesting to me that the results of the ENCODE project have ignited what Carl Zimmer called in an article published in the New York Times, the junk DNA wars. And this is a civil war that's taking place within biology. This is not a war taking place between creation model advocates and ID proponents and evolutionary biologists, but rather this is a civil war taking place between genomic scientists and evolutionary biologists, where genomic scientists, by and large, I think, accept the importance and the significance of the ENCODE project results and find that information useful, whereas evolutionary biologists have derided these results, arguing that, in effect, that if indeed the ENCODE project results are correct, then it means that key features of the evolutionary paradigm simply cannot be valid. And because, according to evolutionary biologists, we know that the evolutionary paradigm is airtight, it means that the results of the ENCODE project must be fundamentally flawed. And this is an interesting argument I think, because it's somewhat of a bizarro world approach to science, where the theory is sitting in judgment of the data as opposed to the data sitting in judgment of the theory. And one of the chief critics of the ENCODE project is a biologist, I think at the University of Houston, by the name of Dan Grahr, who shortly after the publication of the ENCODE project results, published a paper in Genome Biology and Evolution, where he really took the ENCODE project and the results of the ENCODE project to task. And even though this article is somewhat dated now, it's not quite 10 years old, many of the ideas that Grahr presents in this article are ideas that are still in play in terms of criticizing and critiquing the ENCODE project. And so I'm going to step through some of Grahr's criticisms and then offer a response to them. And then from there, we'll continue to look at other types of critiques that have been leveled as well. In a nutshell, there are three major issues that Grahr and others like Grahr have identified with the ENCODE project. The first has to do with the definition of function. The second has to do with the logic of how the ENCODE project established functionality. And then the third has to do with what Grahr claims is a confusion between biochemical activity and function. Now, with respect to the definition of function, Grahr argues that there are two different fundamentally distinct approaches in biology to function. One is what he calls the selected effect. The other is what he calls a causal function definition. A selected effect definition, according to Grahr, refers to a trait for which it has been selected or by which it has been maintained through, again, through natural selection, through selection. So in other words, what Grahr is basically saying is that for something to be, again, functional, you have to understand that functional element in evolutionary terms. And if it shows evidence that that region of the genome has been selected, that it's under the influence of natural selection, then we can consider it to be functional. Otherwise, we can't legitimately consider it to be functional piece of DNA. According to Grahr, the causal function concept is that for a trait Q to have a causal function G, it is necessary and sufficient that Q performs G. So in other words, if we say, for example, that a particular region of the genome is a protein binding region, let's say it binds a transcription factor, that binding of the transcription factor would be the functional role, then it's necessary and sufficient for, again, that Q performs G. So in other words, if you observe that if a piece of DNA is functioning as a transcription factor binding site, and we observe that it binds a transcription factor, then we can conclude that it must be functional in serving a role as a transcription factor binding site. That's the causal function definition. And according to Grahr, only sequences that can be shown to be under selection can be claimed with any degree of confidence to be functional. Now, the first point I have to that assertion on the part of Grahr, because when you adopt that approach, then you would argue that minimally about 10% of the human genome, maximally about 10% of the human genome is functional. The rest is non-functional DNA, according to that definition. But the first point I want to make is that that's a theory-laden definition of function. In other words, that's a definition of function that is predicated on the evolutionary paradigm being valid. And whether you believe the evolutionary paradigm is valid or not, at this point, that's irrelevant. The point is that any kind of definition of function really should be a definition that is independent of any theoretical framework. of any paradigmatic framework. Because as soon as you have a theory-laden definition of function, that again, I think creates a circumstance in which becomes impossible in principle to actually now evaluate that theory in light of that particular observation. So this is not this is a problematic. This is, to me, is problematic with Grar's assertion that function is only for regions of the genome under selection. Now, I would also say that when you go to any other area of science outside of biology, science is about establishing cause and effect relationships. That causal function concept is essentially, it essentially permeates all other disciplines of science. That's what science is about, is trying to establish those causal relationships, and employing a causal function definition of functionality. And in fact, as a biochemist, almost every study that I've ever done involves that approach to functionality, namely a causal function approach. And so, again, this is the point that I just made, that causal function is routinely used in biochemistry and other areas of science. Now, in preparation for the show, as well as in an interaction I had on social media, both Donnie and another person I was engaging brought up this particular article, this perspectives article published in PNAS in 2014, as essentially an article that explored the question, how should function be defined in biology? And they brought this article up as a challenge to the ENCODE project's approach to defining function. However, when I read the article, I found it to be a very thoughtful, very balanced, and very, again, insightful piece on what does function actually mean in a biological context. And what they identified in this article were three different approaches to function: an evolutionary approach, which is similar to the approach that Gar is arguing for, namely that regions of the genome are functional if they're under selective constraints. They also brought up the genetic approach to function, which is to argue that if you have a perturbation in the genome, you look at to see what is the phenotypic effect. If there is a significant phenotypic effect, then that you would consider that particular region of the genome to be functional. And then the last approach would be the biochemical approach, which is what the ENCODE project engaged in, was looking for different types of biochemical activity, where that activity was then assumed to indicate functionality. And what you see is that the biochemical approach tends to give you a very high degree of functionality when you look at the human genome. The evolutionary approach gives you a very low degree of functionality. And so these are, again, definitions that are, if you will, on the opposite ends of the extreme. And in the article, the team that was writing this piece essentially rightly pointed out that each of these approaches have limitations, and each of these approaches have strengths. And they actually argue for kind of a blended approach where all three different approaches are coming into play to assess functionality in the genome. But they highlight problems with each of these. For example, when it comes to, again, looking at regions of the genome as to whether or not they're under selective constraint, again, this is a theory-laden approach to defining function. But on top of that, this approach, I think, neglects the fact that that just because something isn't under selective constraint doesn't necessarily mean that it's still not performing a function. And so I don't think that this is actually a very good approach to defining function. And let me just give you kind of a silly example, because I could argue that, my foot may not have anything to do with my capacity for reproductive, fitness, uh, but yet, you know, just and you could cut off my foot, you could cut off both of my feet, and I would still have again the same reproductive fitness as I would if I had both functioning feet. At least, or I could imagine circumstances where that would be the case. Uh, Yet it's not. It's would be inappropriate to say that my feet don't perform some kind of function. So that's, to me again, a kind of a simplistic illustration, but I think it highlights the limitations of the selective constraint approach. When it comes to the phenotypic effect, uh, this approach again is an interesting approach for function, but again, one of the problems here is that we don't understand the relationship between genotype and phenotype. And so until we have that understanding, it's very hard to use this approach, I think, to establish unequivocally whether or not regions of the genome have function. And if there is any kind of redundancy in terms of genomic function from region to region, this phenotypic effect is not going to uh, you could disable a region of the genome and see no effect on the phenotype and conclude that that region is not functional. But if again that the functionality of that region is redundant, it's where it can be compensated for by other regions of the genome. You couldn't rightly say that that region was non-functional. And then last but not least is the biochemical approach. And one of the points that is made by these research teams is that this is actually a fairly robust approach to establishing function, because when you're measuring molecular scale activity, you're very close to the mechanism uh, by which that region of the genome is actually operating. You're actually delving into the mechanistic operations, and so you're very close to having a fundamental understanding of the structure-function relationship between those regions of the genome and again their functionality. And so this is actually a robust way of gaining insight into function, much more so than the other two approaches, which give you no real insight mechanistically as to what is the basis for function. So I think the ENCODE project, in other words, made the right decision to use a biochemical approach, because it does give us information that leads to a mechanistic understanding, a fundamental understanding. Now, another criticism leveled by Gurar is the logic that the ENCODE project employed. He accused the ENCODE project of affirming the consequent, which is a logical fallacy. And so, in other words, if P then Q, we observe Q, therefore P must be true. This is called affirming the consequent. Now, I would argue that affirming the consequent is a problem if you're dealing in deductive reasoning. But in science, we're dealing in inductive reasoning. And in fact, we engage in this kind of reasoning all the time, because it's not a logical fallacy to do that in science when you reason this way, because you're reasoning inductively, not deductively. And in fact, and so what Gurar would argue is this, that DNA segments with a particular function F display property P. Property P is observed, therefore the DNA segment has function F. So if we say that transcription factor binding sites will bind transcription factors, we observe transcription factors binding to a particular site, therefore we assume it's a transcription factor binding site. That would be, according to Gurar, affirming the consequent. But in inductive reasoning, that is actually a legitimate way to, again, think about phenomena, to work towards establishing cause and effect relationships. Because again, there's a difference between inductive and deductive reasoning. And in fact, nearly all scientific measurements ultimately affirm the consequent, because they're done inductively. What that means, though, is, of course, that we have to do additional work to affirm what we think to be the case. So if we think a region of the genome is a transcription factor binding site because it binds transcription factors, we need to do additional experiments to confirm that result. But that type of reasoning is not a flawed reasoning. It's not flawed logic. The ENCODE project employing that type of reasoning was well founded. And by the way, Groar affirms the consequent when he argues for a selected effect definition, where he argues that, you know, if if a region of the genome is under selection, it must be functional. We see a region of the genome under selection, therefore it's functional. That's affirming the consequent. And so Groar is guilty of that same kind of logical fallacy as the ENCODE project is. Now, to me, the criticism that Groar raises here is actually legitimate criticism, and that is the ENCODE project measured biochemical activity and assumed that activity meant that it was functional, that they ascribed activity or function to activity, to biochemical activity. And it's quite possible that some of that activity was simply spurious, that some of that activity was essentially biochemical noise. And I think this is something that is a legitimate concern that's being raised. However, when I saw Groar raising this concern, almost 10 years ago now, my response at that time was that I was skeptical that the activity measured by the ENCODE project was largely noise as opposed to reflecting functionality, for two reasons: for energetic considerations, as well as for the concerns about whether or not you would have an interference problem if that level of activity was actually biochemical noise. And so let's kind of step through each of these in turn. So when it comes to transcription, I think about 60% of the genome, human genome, is transcribed, if I'm not mistaken. And if that's the case, and much of that transcriptional activity is biochemical noise, that to me would be deeply problematic, because transcription is a energy-intensive, resource-intensive process where you're utilizing essentially, nucleotides. You're utilizing nucleotides, which are valuable resources for the cell, to produce RNA molecules that, again, are just simply the result of biochemical noise. Plus, it takes 14 kilocalories per mole every time you add a nucleotide onto a growing polynucleotide chain, whether it's RNA or DNA. And that's a very energy-intensive process. And so because you're using up valuable resources, because you're using up large amounts of cellular energy to produce transcripts, if they are non-functional, it seems to me that even from an evolutionary perspective, that kind of wasteful process would actually be minimized. There would be mechanisms in place and very strong selective effects in place that would essentially try to dampen that kind of unnecessary transcription. And it turns out that when we evaluate the transcripts, that many of these transcripts actually, again, of the 60% of the genome that are transcribed, seem to have functional utility. 2% of the transcripts are the protein coding genes, 27% would be regions that are transcribed but not translated, and these would be coding associated. So this would be things like introns, for example. There's also regions that are transcribed but not translated that are non-coding regions. And then pseudogenes are also transcribed as well. And when we look at the list of the transcripts, not only do we see, again, messenger RNAs that reflect protein coding regions of the genome, but we also see transfer RNAs, ribosomal RNAs, nucleolar RNAs, spliceosomal RNAs, microRNAs, small interfering RNAs, and long non-coding RNA molecules. And all of these that we see are known to have function. In fact, a few years ago, a research team looking at non-coding RNAs basically argued that most transcripts play some kind of functional role. That non-coding RNAs are not simply transcriptional byproducts or splicing artifacts, but comprise a diverse population of actively synthesized and regulated RNA transcripts. These transcripts can and do function within the context of cellular homeostasis in human pathogenesis. And the point here that I'm making is this: that this is a research team that is working independently of the ENCODE project and has concluded that the transcription that we see in cells is actually mostly functional, that it's not biochemical noise, and that this is essentially giving legitimacy to the results of the ENCODE project. Now, this brings us to the point where I want to talk about the competitive endogenous RNA hypothesis. This is an idea that essentially explains the functional role of duplicated and unitary pseudogenes, but also explains the role that long non-coding RNAs play, as well as a role that microRNAs play also. And the idea behind this model, which is actually gaining some strong experimental support, is that RNAs actually are transcribed as families of RNA molecules. That transcripts form a family of transcripts, and there's different families of transcripts, and that these different families of transcripts essentially are regulated in concert with each other. And what defines a particular family of transcripts would be the microRNA binding sites on the transcripts. And so what happens is you have RNAs that are transcribed that have, again, the same microRNA binding sites. When these microRNAs bind, let me go back here. When these microRNAs bind to the transcripts, they essentially target those transcripts for being destroyed by the RISC complex. And so this destruction means that when RNAs are basically being transcribed, they also are being targeted for destruction. So it's kind of like a biochemical feudal cycle where RNA is being produced and then is being destroyed. And so this is actually a type of gene regulation. So it turns out that not only is gene regulation affected by whether or not a transcript is being produced, but also once it's produced by whether or not it's destroyed before it has a chance to be translated at the ribosome into a functional protein product. And so the idea here is that you have these, again, microRNA sites that create families of RNAs, families of transcripts. And so if you upregulate the transcription of one of members in that family, they essentially are going to function as molecular decoys that will lead to their destruction. And as a result of that, they will allow other members in that RNA family to actually be translated at high levels. And so this is a role that is being played by not only long non-coding RNAs, but also by unitary pseudogenes and by duplicated pseudogenes as well. And in fact, duplicated pseudogenes actually are playing a decoy role in terms of gene regulation. But the point here is that, again, you have these RNA families in which the translation of members in that family are dependent upon, again, the levels of other transcripts that are part of that family. And that you have, again, this network of of RNA molecules that, again, are transcribed and translated in concert with one another, where again, this decoy mechanism is at play. And so this explains in part why you have long non-coding RNAs and other types of RNA molecules being transcribed. They're playing an important role in gene regulation. Now, another criticism that we see that's kind of related to this idea of transcription is that some of these transcripts are produced at very low levels and therefore, again, must be spurious, must be biochemical noise. But this is not necessarily the case, because just because a transcript is, again, being produced at low levels doesn't mean that it's actually not having a very important and very impactful regulatory effect. Just you can't necessarily correlate the level of transcription with whether or not it's functional or whether or not it's biochemical noise. Now, another concern I have with this idea that activity is not functionality, but it's biochemical noise, is that, again, if you're creating, this has to do with primarily the transcription factor binding sites, is that you would basically have a biochemical interference taking place if you're producing RNA transcripts that are randomly binding to sites in the genome. And so, for example, it's not unreasonable, I think, first of all, to ask the question, is transcription factor binding actually noise, or is it actually functionality, or displaying functionality? Because, most transcription factor binding sites are about 4 to 10 base pairs in size, and there's about, typically about 100 binding sites per genome for a particular transcription factor. But when we look at the genome, which is 3.2 billion base pairs in size, there could be anywhere from 1 million to 1 billion potential binding sites for that particular transcription factor. Because you could statistically have that binding site recur by accident in a genome that's 3.2 billion base pairs in size. You could have variants of that transcription factor binding sequence that would be potentially high affinity binding sites, other variants that would be medium affinity, and then some variants that would be low affinity binding sites. But the point is that you could have, in principle, statistically speaking, a large number of potential binding sites. But a study done and published in 2016 indicated that that high affinity non-functional binding sites in the human genome and the genomes of organisms really across the board are extremely rare. In other words, they actually measured transcription factor binding sites affinities, and then through statistical analysis showed that high affinity non-functional sites are rare in the genome, meaning that most of the binding that was measured by the ENCODE project was probably functional binding. And there's a good biochemical reason for why this is the case. The first has to do with this interference effect. That is, if transcription factors were binding randomly all over the genome, that would just simply muck up the operation of the genome. If most of the binding was random binding to regions of the genome, that would create an interference that would gum up the operations associated with the genome. There's also a titration effect. In other words, there's only so many copies of a transcription factor that are going to be produced. And if these transcription factors were binding randomly to different sites in the genome, then there wouldn't be any of those transcription factors available to actually do the binding that's needed for these transcription factors to properly function. And so, in other words, there are very good biochemical reasons to think that transcription factor binding is not noise, but is actually reflecting functionality. Now, this leads us to really what is the real reason that people like Dan Grahr are actually challenging the ENCODE project. And this is what John Maddock has said. a molecular biologist. There may be another factor motivating Gurar and others, which is suggested by the sources and selection of quotations used at the beginning of the article, as well as the use of the phrase evolution-free gospel. The argument of largely non-functional genome is evoked by some evolutionary theorists in the debate against the proposition of intelligent design of life on Earth, particularly with respect to the origin of humanity. In essence, the argument posits that the presence of non-protein coding or so-called junk DNA that comprises greater than 90% of the human genome is evidence for the accumulation of evolutionary debris by blind Darwinian evolution, and argues against intelligent design, as an intelligent designer would presumably not fill the genetic instruction set with meaningless information. The argument is threatened in the face, this argument is threatened in the face of growing functional indices of non-coding regions of the genome, with the latter reciprocally used in support of the notion of intelligent design and to challenge the conception that the natural selection accounts for the existence of complex organisms. And so, in other words, the reason, again, for challenging the ENCODE project results has more to do with really the threat of intelligent design, and the fact that the ENCODE project result actually threatens to undermine key ideas in evolutionary theory. In fact, I did a debate with P.Z. Myers a few years ago at North Dakota State University on Darwin Day, and the idea of the ENCODE project came up. And P.Z. Myers said to me that if the ENCODE project results are correct, then it means that there are key ideas in evolution that can't be true. And so therefore, the ENCODE project results simply must not be correct. And so this highlights really what's at stake in the mind of many people. And then again, I just want to close out by just simply pointing out that in addition to the ENCODE project, there are a mounting number of studies that consistently indicate that, again, a junk DNA has function. We now know that pseudogenes can code for functional proteins in some instances, and they can, again, function in a gene regulatory role through a decoy mechanism that is part of this competitive endogenous RNA hypothesis. And this, by the way, explains why pseudogenes look like the corresponding version of genes that produce produce protein products, is because their role in this regulatory mechanism is predicated on their similarity to pseudogenes. Now, I've had people tell me that, well, this explains functionality for pseudogenes that are transcribed, but what about the pseudogenes that aren't transcribed? With the assumption that many of the pseudogenes in the genomes aren't transcribed. Well, it turns out that assessing whether or not a pseudogene is transcribed is actually a very tricky thing, because there are researchers that are now arguing that the assays used to measure transcription may be mistaking pseudogenes for the functional version of the genes because of sequence similarities. So I don't think it's established that many pseudogenes are actually not transcribed, even though that, again, is the claim. Other classes of junk DNA are also functional. Line DNA plays a role in gene expression, as does sign DNA. Introns allow for alternate splicing, and they also seem to be playing a role in stress response. ERVs and EVEs play a role in have an antiviral function. They're part of the innate immune system. Tumor suppression is also something that ERVs play a role in, and elements of ERVs and EVEs play a role in gene regulation. And there's also other roles that junk DNA plays, quote unquote, that have nothing to do with an informational role, but a non-informational role. there's something known as the nucleoskeletal hypothesis that we'll talk about in a minute. Well, we'll go ahead and talk about it right now. But the idea here is that the DNA content in the nucleus actually determines the nuclear volume, and the ratio of the nuclear volume to cellular volume is a tightly constrained parameter. In other words, as the cell gets bigger, the nucleus has to get bigger in proportion, because as the cell gets larger, you have to have the ability to supply basically transcripts and other materials from the nucleus in order to sustain the activity of the cell, which requires a greater surface area of the nucleus and a greater number of nuclear pores. And so you have a very tight relationship between nuclear volume and cellular volume. And this is actually determined by the DNA content. So in other words, some of that this nucleoskeletal idea, this hypothesis, could actually explain the C-value paradox in a functional role, in a functional way, as opposed to it resolves the C-value paradox by ascribing function to DNA, to non-coding DNA. Here's a couple of diagrams. On the left, it shows the relationship between nuclear volume and cellular volume, and you can see that that ratio has to be very, very tightly constrained. Between about 30, the nuclear volume has to be about between 30% and 10% of the cellular volume in order for again, organisms to survive. And then we also see on the right-hand side, again, the correlation between nuclear DNA content and cell volume, showing that some of the so-called non-coding DNA could be playing a nucleoskeletal role. And again, this resolves the C-value paradox in a way in which you're looking at that non-coding DNA as being functional as opposed to being the random product of an evolutionary history. In addition to that, there's some people that have argued that junk DNA may actually serve a role as a mutational buffer. Others argue that it actually plays a role in maintaining the three-dimensional structure of the genome. And this leads to a recently discovered concept known as the genome code, which basically argues that a number of the non-coding sequences in the genome are actually playing a role in helping to essentially define and establish and maintain higher order three-dimensional structures of chromosomes, which we're now learning is absolutely critical for the function of those chromosomes, in part because that three-dimensional structure is necessary to bring enhancer sequences close to the gene that is going to be transcribed and translated. These functional elements are pervasive throughout the genome, and in fact, this structural code overlaps the genetic code. The scientist Bernardi, who proposed this idea, writes, Ono, mostly focusing on pseudogenes, proposed that non-coding DNA was junk. Doolittle and Sapienza and Orgel and Crick suggested the idea of selfish DNA, mainly involving transposons visualized as molecular parasites rather than having an adaptive function for their host. In contrast, the ENCODE project claimed that the majority of the genome participated in at least one biochemical RNA and or chromatin-associated event in at least one cell type. At first sight, the pervasive involvement of isochores in the formation of chromatin domains and spatial compartments seems to leave little or no room for junk or selfish DNA. Okay, and so in other words, let's see here, let me stop sharing. In other words, this idea of most of the genome being functional is not only an idea that I think is established by the ENCODE project, but there's other independent studies that are, studies independent of the ENCODE project that are reaching comparable conclusions, that are affirming the results of the ENCODE project. And to me, the criticisms that have been leveled against the ENCODE project for the most part, don't stand. For the most part, I think lack real rigor or lack any kind of real legitimacy.
Well, that was another fantastic presentation, Dr. Rana. I loved all your points, especially your assessment of the paper defining functional DNA elements in the human genome. I also appreciate the fact that you addressed so many of the criticisms, including this idea that low-level transcription or low-level activity must mean that the activity is spurious. And you also address pseudogenes, ERVs, and so much more. So I'd like to point out, though, that from the genetic archetype starting point and model, right, Dr. Rana, that you're proposing, you can make some really valid predictions pertaining to the function of these DNA elements, such as pseudogenes and ERVs. So the design hypothesis would then predict function, while from my understanding, the evolutionary model would predict much non-functionality. And since many of the lines of evidence used to support common ancestry, Dr. Rana, are then agnostic to the debate, as you have pointed out, the genomic archetype or common design model can equally explain the data when it comes to those lines of evidence. Therefore, my question is, could DNA function be a plausible way to help differentiate the models and tell us which one is right and which one is wrong?
Yeah, I mean, that's a great question. And, from my perspective, at least at this point, I think we're looking at, what you might say would be, in effect, a standoff. In other words, I think, the first step that we need to take as people that are ID proponents that are, that advocate for kind of a creation model perspective is to simply establish the fact that a common design approach has equivalency or is an equally valid explanation as is common descent. Because so often I hear people say that the only way to explain the nested hierarchical structures of non-functional DNA is through again common descent. But if we can again show that these sequences are functional, and we can show that there's a rationale for why these sequences bear similarity to, let's say, viral genetic material, or why pseudogenes display similarity to genes, then I think we can actually come to the table with a comparable model. Now, I at this point, the question you're right asking is the right question: is how do we distinguish between these two models? And that's, that's something that I need to give a little bit more thought to. But I think by virtue of the fact that you have statements by people like P.Z. Myers, or you have people concerned about the C-value paradox being undermined by the ENCODE project results, since those are actually key, ideas in the evolutionary framework. I think the fact that they're threatened by the ENCODE project result actually tilts the scales in favor of kind of an ID approach. Something else too that I think we can make by way of prediction. And by the way, I just want to point out that in 2000, one of the very first articles I wrote at Reasons to Believe made the prediction that we would eventually discover function for virtually every class of junk DNA. This was a prediction made, again, 20 years ago, and is essentially being satisfied by fairly recent results. And I wasn't unique in making that prediction. I know a number of ID and advocates and creationists making comparable predictions. So this is a prediction that I think we want to make sure we're getting credit for at this point. But as we go into the future, what I'm noting is that our understanding of the genomes of organisms in general is in its infancy. And I suspect that when we start looking at the complexity of the gene network interactions that are taking place, that we're probably going to have to develop a systems biology approach to really understand what's going on, that it's going to become, genomics is going to become the domain of engineers, not the domain of biologists. And what I predict is that, we're going to discover that not only is our engineering principles necessary to make sense of what's genomic function, but that we're actually going to discover principles in essentially the regulation of the genome that is going to inspire new approaches to systems biology. In other words, I'm predicting that that the designs that we see in genome operations are going to be so sophisticated and so impressive that it's going to inspire new technology. And so that would be a prediction that I would make now, that I think is something that I don't know that you would necessarily make from an evolutionary perspective, or at least would necessarily flow from an evolutionary perspective.
Amen. Well said. That is a fantastic prediction. So many good points you made there, Dr. Rana. It's hard to choose which one to touch on. And from my understanding and from looking into the literature, the evolutionary community has at least assumed lots of junk or evolutionary leftovers, as in the genetics of living organisms should reflect universal common ancestry. So they should find genomic fossils, and therefore they look to pseudogenes. As you pointed out, the ancient remnants of past viral infections with the ERVs and ALU sequences and so on. But I've never actually seen them, prior to discovering the incredible functionality of a lot of these DNA elements, I've never seen them make the point that this is expected or this is predicted. It's kind of like when we find this data, they retrofit it in somehow. Have you found that they expected this or even made these predictions prior to finding just how functional a lot of these, let's just say, ERVs are?
I have a similar experience as yours, where I see very little in the literature where there were any predictions about function for any of these different classes of junk DNA prior to the discovery that they indeed have function. And one criticism is that, well, what you're doing is you're taking the discovery of functionality for a subset of a particular class of junk DNA as evidence that the entire class of junk DNA, all these types of junk DNA elements are functional. But the fact of the matter, it's only really a small portion or a small fraction. But this assertion is actually being undermined as we do more and more work, and as we're discovering, things like the competitive endogenous RNA hypothesis, because this is now moving from, a particular sequence has just acquired function through some kind of neo-functionalization, to actually saying, no, we actually have an elegant theoretical framework that explains why all of pseudogenes are actually functional, right? And same things with understanding endogenous retroviruses and EVEs as being part of the innate immune system. Now we have an elegant explanation for why these sequences look like viral elements, and we're seeing that more and more data is indicating more and more pervasive function for the different classes of junk DNA. It's not just an isolated case here or there. And that to me argues against neo-functionalization, where I would expect if the functionality is rare, then you could say neo-functionalization makes sense, where evolution is just using the material that's laying around and co-opting it and modifying it to create systems with new function. But if the function becomes pervasive, then this neo-functionalization argument becomes harder and harder to make, I believe.
Right, exactly. Because if what we see based on the prediction is genome-wide functionality, which the trajectory seems to favor genome-wide functionality, including these DNA elements like the ERVs, the pseudogenes, ALU sequences, then to employ neo-functionalization or co-option for such pervasive functionality in the genome, that really seems kind of like a stretch, right? Because it's
right. And to me, I think this is a good place to bring up this point too, is that many times evolutionary biologists simply declare regions of the genome to be non-functional without actually doing any kind of experimental work.
Right.
It's all based on what they expect to be the case, given evolutionary theory, right? And so, theory guides, experiments decide, and to me, nobody, in many instances, when again sequences are declared to be non-functional, people have not done the experimental work to show that's the case. It's just rather, again, an assumption that's made.
Well said, Dr. Rana. Well, here's an argument, and I appreciate how thorough you were in your presentation, because you really nailed it on a lot of these criticisms raised against ENCODE. But another argument is, they'll say, because approximately 5% of the human genome is conserved across all mammals, the critics will say that since conservation is strongly indicative of function, the biochemical function that is outside of the conserved regions must then be non-functional. Have you heard this argument, and what would be the best way to respond?
Yeah, I have, and in many respects, that argument is, in a sense, very similar to the argument that Dan Grahr was making about the selected effect definition of function.
Right.
That is, in other words, if you can't show that a region is under selection, then you can't really conclude that it's functional. And so that 5% number would be a number that, Grahr, I think, argues that the maximum level of functionality reasonably in the human genome would be on the order of 25% under extreme conditions. And he argues that most reasonably it's probably about 10%. And so this is, if it's selected, then it's going to be a region that is presumably conserved. It's a comparable idea that the conserved sequences are essentially those sequences that are under selection, and they're not able to change because they're of critical functionality. But just because you have non-conserved sequences doesn't mean that they're not functional. You just simply can't conclude that. It could very well be that they may be regions of the genome that actually can tolerate high levels of mutational change and still retain their function, particularly if those regions are actually playing a non-informational role, like, let's say, part of the nucleoskeletal role, or if they're playing some kind of role as a mutational buffer, as an example. Or maybe there are regions of the genome that can tolerate high levels of changes and still retain function. So, for example, regulatory elements typically can undergo a greater degree of mutation without compromising function than protein coding regions of the genome. We also are making the assumption that those differences are due to evolutionary changes. Mutational changes. It could very well be that they are intentionally designed in that way because they're performing different types of functions, though maybe related functions, but they may be functioning a little bit differently in different organisms. And so, is that lack of conservation reflecting selection, some kind of selection effect, or is it actually intentionally designed that way because it needs to be different in order to perform the specific function it performs in that particular organism? So I don't think you can necessarily conclude just because something is non-conserved that it's not functional.
Great response. So many good points. And that's my thinking as well. Firstly, as you pointed out, they assume that the conserved sequences are important and functional, and therefore selection's preserving them over time, right? But as you've pointed out, the non-conserved regions can certainly be there for a number of reasons: backup code, or playing a nucleoskeletal role. And also what you kind of said there at the end, and I wonder if you would agree with this, if we as intelligent design advocates would propose special creation events, then would we then predict that some non-conserved regions are maybe just specific to different creatures that maybe aren't related, and therefore their functional roles will be specific to different creatures?
Yeah, that exactly. I would agree with that. That's definitely a possibility.
Yeah, that's a great answer. I appreciate that because, Dr. Rana, I hear that argument over and over and over again. So I'm kind of glad that we touched on it there a bit. Now, I want to point out, I've read so many of your articles. I highly recommend them. And on November 3rd, you published an article titled Endogenous Retroviruses Help Fight Tumors. And I love the way you ended the article, Dr. Rana. You pointed out that so far from being the villain of this tale, ERV sequences turn out to be P53's faithful sidekick in the battle against cancer. Can you elaborate a bit more on this article and why it's important in terms of this debate we're having?
Yeah, well, P53 is considered to be, kind of the guardian of the genome, where it's playing a number of roles that help to maintain genomic integrity. And one of the roles that it plays is actually suppression of the transcription of endogenous retroviral elements in the genome. But it turns out that when the cell is actually under stress, uh, the proteins that work in conjunction with P53 become inactive, and this actually leads to P53 causing the transcription of endogenous retroviral elements. And what ends up happening is when these ERV elements are transcribed, it gives the cell the appearance of actually being invaded by a viral invader, having a retroviral infection, which then targets that cell for destruction by the immune system. And so it turns out that there are some tumor cells that actually can evade detection of the immune system. And so this mechanism that's mediated by P53 is a way for those uh, tumor cells that are evading the immune system become now present, or now become detectable by the immune system, so that the immune system can clear those tumor cells out of the body. And so researchers are looking to take advantage of this fact to actually develop these P53 uh, in, these drugs that will essentially stimulate P53 activity, and specifically the transcription of ERVs as an anti-cancer therapy. But in other words, what we're looking at here are these endogenous retroviral elements actually playing a role in tumor suppression uh, by creating by essentially viral mimicry. That's I think the phenomena given, or name given to that phenomena, is viral mimicry. And so this is just, really cool discovery that shows us that, we really have misunderstood, what endogenous retroviruses are doing in cells, and that they actually, again, are playing very important functional roles. And just because, again, they're not normally transcribed doesn't mean that those ERV sequences aren't actually critical,
Right.
because normally they wouldn't be transcribed. And that would be, again, something that people would point to, saying, look, this indicates that they are indeed non-functional. But their transcription uh, will happen only under select circumstances. But under those circumstances, that they are indeed functional. And so you can't just simply conclude because a sequence isn't transcribed, or it's transcribed at very low levels under certain cellular conditions, or for certain cell types in certain tissues at certain stages in development, that these sequences are necessarily non-functional. They could very well be functional only for brief periods of time in certain cell types under certain conditions.
Now, so much amazing information. So it's safe to say then, Dr. Rana, that these two main arguments that uh, critics of intelligent design and critics of the ENCODE project, for example, how they say that, well, low level transcription or low level activity means the activity must be spurious or just noise. And then at the same time, they look to the non-conserved region. So they'll say, okay, low level activity and non-conservation means it's probably not really functional. But as you're demonstrating here, no, those are not two good criteria to determine whether something is functional or non-functional.
Right. I mean, the ultimate criteria is that you have to roll up your sleeves and do the hard experimental work,
Right.
where you need to really probe, whether or not indeed it's, those sequences are functional or non-functional.
Great response. And I apologize if some of this you addressed in your presentation because it was so thorough. But as you know, the critics will point out that this transcription, which they'll say is 60+% of the genome being transcribed, they'll say about 2/3 of this transcription is exons and introns. And since introns are non-protein coding and would make up the greater portion of that, they'll point out that introns are transcribed and then cut out of the RNA before translation occurs, right? And they will admit, from my interactions with them, they will admit that parts of the introns are functional in the sense that they serve as markers at their ends, but they'll say that the long regions, Dr. Rana, in between, the cut here markers are very messy, and they'll point to, the fact that they're full of transposons and non-conserved random sequences, and they'll say, so therefore, the majority of it is still non-functional, even if we have a little bit of a function on the ends of them. Does that make sense to you, or, what would be a good way to respond to this?
Yeah, well, and first of all, I would just say that the presence of introns, in genes actually allows for alternate splicing, allows for that transcript to be modified in a variety of different ways to produce a number of different alternate transcripts, and so one gene can actually, in effect, encode for several different types of protein products. And so when you have alternate splicing, the introns that are not removed are actually playing a role in the structural makeup of that particular protein product. Number one. Number two is that there have been studies published recently that have shown that introns that are spliced out of genes actually can go on and play other regulatory roles, in the cell. So they're not necessarily just simply RNA discards, but they actually are playing other regulatory roles, particularly playing a role in the biochemistry of the stress responses during conditions like starvation and things like that. So we're again just assuming that introns are non-functional, that they only are serving one particular role in biochemistry. And the fact of the matter is they may be doing things that we've yet to discover. And so this is really there's not experimental evidence that they're non-functional or unimportant. It's essentially an assumption being made. Whereas on the other hand, we're seeing other studies actually indicating that there's a wide range of different types of functions that introns actually assume.
I love it. It's such a great time. It's 2021, and it's so much fun because we're, as you know, I mean, there's just so much information, so many more papers and data coming out showing just how, functional portions of the genome are that were once assumed to be non-functional. As you pointed out, one gene can encode for several different protein products. And I find alternative splicing fascinating, and where the evolutionist says most of the introns are just RNA discards, as you've put it, Dr. Rana, they actually indeed do function in a variety of ways. And also we're in the infancy of understanding the DNA language in the first place, right? And that's why, as you've pointed out, we can make predictions and we can be confident about those predictions because of the trajectory, where I'm not seeing, the evolutionary community really making predictions. I guess they're just more so attacking, the ENCODE project and just hoping for junk.
Yeah, and to be clear, there are a number of life scientists who actually do see enormous amount of value to what the ENCODE project has done, right? And the ENCODE project has, again, put together an impressive collection, you know, of you know, or impressive catalog of biochemical activity that's associated with different regions of the genome, that then provide a starting point, I think, for people to really begin to understand, you know, individual, you know, elements of the genome and whether or not they're functional and what that functional role might actually be. So, you know, so there are scientists, to be fair, that really do see value to the ENCODE project.
Well said. Well said. Yeah, I should be more clear on that, in that we do have a lot of critics that kind of fight and attack it. But at the same time, there are a lot, and from my understanding, a lot of scientists that are involved at the NIH, National Institute for Health, that do see a lot of good out of these research programs. Here's a good question, actually, Dr. Rana. What are your thoughts on the gulo pseudogene? As we have talked about pseudogenes and these so-called genetic mistakes, but specifically the gulo pseudogene. Do you find this to be convincing evidence for human evolution and ancestry with the great apes, and kind of how do we deal with this?
Yeah, and you know, to me, the when it comes to the gulo pseudogene, the way I would respond is that the competitive endogenous RNA hypothesis essentially explains a functional role for unitary pseudogenes. Is that even though, you know, these pseudogenes may no longer produce uh, or may not necessarily produce you know, transcripts that are translated. Oops,
No problem.
sorry about that.
No
Even
worries.
though they may not produce transcripts that are translated, they still are producing transcripts that are part of this this microRNA network of transcripts, and so their activity in that network is influencing the translation of other transcripts that are part of that network. So they're playing a regulatory role, even if they're not necessarily playing a role in which the information is transcribed. Sorry, is translated. So yeah, so you know, I don't know what specifically the function of the gulo pseudogene is necessarily, but its generalized function would be part by virtue of the fact that it's playing a role
Right.
in this network.
Right. Great response. Great response. Here's a question as we start kind of winding it down. Time always flies by with you, Dr. Rana. You give us so much great information, and a lot of people in the chat, and I feel the same way, are pointing out that this your presentation's rewatchable. You know, it's going to have to require a few watches to kind of soak all the information in. This question came in from Guzman 1611. I appreciate the question, Guzman. He says, question for Dr. Rana. How should we as creationists or intelligent design advocates define information from a biological point of view?
Wow, that's a really good question. And you know, I think that there are two different concepts of information that I think would be that are practical, or sorry, that are helpful. One would be defining information in semantic terms, and so you know, this would be related to kind of the idea of Shannon information, where Shannon information is just simply the probability of getting a particular sequence of elements or characters in a particular order, where the lower the probability, the higher the information content. But you can have Shannon information that has no meaning whatsoever. What is important when it comes to information in a biological context is the semantic concept, where there are particular sequence combinations that actually harbor biochemical functionality and hence biochemical meaning. And what gives meaning to syntactical information would be the genetic code. So you can't really think about semantic information until you're starting to think about really the genetic code and the types of protein products that are being essentially that are essentially being defined, if you will, by that information found in DNA. So when you start talking about semantic information, now you're starting to talk about kind of an integrated set of systems that's giving meaning to that information. Another different approach that I think is helpful would be an algorithmic definition of information, where the algorithmic definition would be what is the set of instructions required to produce that particular sequence. So, sequences that have high levels of algorithmic information are sequences that require a very complex set of instructions to actually account for the sequence, where the maximum amount of algorithmic information would be essentially the set of instructions that essentially is a delineation of the actual sequence itself. But one of the things that's really provocative is that it seems as if when you look at biomolecules, is that there's a set of algorithmic information that's instantiated within the structure of biomolecules, that set of instructions, again, is part of an integrated system of biomolecules, where that information or those set of instructions seems to tell those molecular systems how to essentially function in conjunction and collaboration to accomplish some kind of end product. And so those are two concepts, I think, of information that are kind of distinct from each other, but are very helpful. I personally think the algorithmic information concept is the most useful and the most versatile, and also the one that I think raises the most significant challenges to an evolutionary explanation for bioinformation.
Great response to a great important question, Dr. Rana. Let's end it with this next question. And I love that because I'm looking over my notes, and a lot of the criticisms that have been raised against ENCODE and arguments in support of junk DNA, and we've touched on them all. You've done a great job addressing them. Again, another one of your articles, though, that I really enjoyed, Dr. Rana, was published on October 20th, 2021. It's titled "Endogenous Viral Elements, EVEs, Help Wasps to Parasitize." And in the article, you provide a response to the problem of natural evil and to bad designs. I was wondering if you could touch on this important issue, even just briefly, Dr. Rana.
Yeah. Well, I mean, this, the idea of junk DNA as a challenge to the design argument is really kind of an embodiment of the problem of natural evil to some degree, which is essentially, why would God create a world that looks this way? If God is all powerful, all knowing, and all good, then we would expect a world that's very different than the world that we live in. Why is there so much pain and suffering in nature? Why is there parasitism? Why is nature red in tooth and claw? Or why do we see flawed designs in biological systems? These kind of systems, the argument would be something we'd expect evolutionary mechanisms to create, not the God of the Bible, who again is all powerful and all knowing and all good. But really what's missing in that assessment is that maybe there's actually a rationale for why the world is indeed the way that it is. And there are very good reasons why parasitism exists in nature. It's a way to control numbers. And so for many of the organisms that are the prey of wasps, that lay their eggs in the bodies of caterpillars and things like that, they're actually playing a role in curtailing the numbers of these pests that would destroy plants, that would destroy agricultural crops. And so you could actually see a reason why God would create a world with parasitism. And what's interesting is that these parasitic wasps actually employ endogenous retroviruses as a means by which to stave off the the response of the caterpillar to the invasion of the wasps laying eggs within its body. The caterpillars will mount these immune responses against the caterpillar eggs, And so what these endogenous viral elements in the genomes of wasps do is actually become activated during the invasion and essentially produce proteins that essentially inhibit the response of the caterpillar. And so it's here that the endogenous viral elements are functioning like a viral vector that are carrying genes into the caterpillar that again are shutting down its innate immune system. But what's intriguing to me is that what these wasps are doing with endogenous viral elements is very similar to what is being explored in gene therapy today, where people are looking at the use of, you know, uh, basically viral vectors as a way to deliver genes to cells for gene therapy. In fact, if you look at the Johnson and Johnson vaccine, the AstraZeneca vaccine, they're basically using an adenoviral vector to carry the gene that encodes for the spike protein into cells in our bodies. And so that's essentially the same kind of mechanism, interestingly enough, that these wasps are using with these endogenous viral elements. So to me, very intriguing.
Very interesting. I appreciate that detailed response, Dr. Rana. Here's a good way to end it, going right back to the main topic of tonight: junk DNA, DNA function, ENCODE, and even predictions pertaining to the evolutionary model and the genomic archetype model. Dan Straight, thank you for the question. He says, how confident are you, Dr. Rana, in the functionality of the quote unquote junk? And what percent would you consider functional overall? God bless you. Great presentation.
Yeah, that's a great question. Number one, I'm very confident in the fact that junk DNA is functional because, again, the trend line continues to move explosively towards functionality. It's in if I'm going to be honest, in 2000 when I made the prediction that junk DNA would be functional, I made that prediction based off of essentially what are the creation model that we were developing at Reasons to Believe would demand. But was I confident that that prediction would be satisfied? I don't know how confident I really was. But here we are 2 decades later, and we now are seeing functionality for things like pseudogenes. We're seeing functionality for endogenous retroviruses in ways that I don't know that I could necessarily have imagined a couple of decades ago. And this recognition that endogenous retroviral elements are part of the innate immune system is something that really in the last 5-6 years has been recognized by biologists, and now there's an explosion of papers essentially going down this path, providing example after example after example of ways that ERVs contribute to the innate immune system. And so I'm completely confident that we're going to continue to see more and more discoveries of function for junk DNA. I think we're going to see more and more studies that continue to affirm the results of the ENCODE project. We're going to see more and more instances where the results of the ENCODE project are proving to be extremely valuable for biomedicine. But what is the exact percentage of the human genome or the genomes of organisms that are functional? I don't know. I think I would suspect, even as a creationist, that there's going to be some level of non-functionality in genomes. I think that's just serves to reason, regardless of your view of how genomes have come to be. I think there's going to be some level of non-functional elements, but I don't know the precise number. I'm really letting the data make that determination. But I think at this point, what these discoveries have done is really change the way that we're thinking about the genome. It's far from being this wasteland of junk. It looks like an incredibly elegant, incredibly sophisticated system that, again, is going to be a challenge for systems engineers to truly to decipher in terms of its functionality.
Great answer. I completely agree. Dr. Rana, so many good points, so much good information. Again, I appreciate your time. I know how busy you are. And, I thank you so much for giving us your time for what I would call this two-part series. We did part one a couple months ago. We did this one. Anybody who has not yet seen part one, please check it out. I've got it on our website, actually, right on the homepage and in the description box of this video. Last time we even dealt with things like hierarchies in genetics. We dealt with third position codon variation, ERVs, pseudogenes. So we've really touched on a lot. So I want to encourage people to please share around these videos. The truth is important. Dr. Rana, again, thank you so much. I appreciate it. Any final words, final thoughts, anything you wanted to add before we kind of wrap it up here?
Well, first of all, Donnie, thanks for having me. You've been a gracious host, and I'm, honored to be, part of what you're doing at Standing for the Truth. So thanks for having me. And, I think the if there's a final thought, it would just simply be this, and it's a point we've already made, but just keep in mind that we really are very much in our infancy, excuse me, when it comes to studying genomes. And I think we want to be just a little bit circumspect before we start drawing conclusions, until we actually get a better understanding, a better grasp of these systems. But, I also think that what we're seeing is that a creation model approach, an intelligent design approach to genomes actually is a very fruitful way of thinking about them. It leads to advances, it leads to discoveries, and I would dare say that an evolutionary paradigm is actually stymied and stultified advances in genomic science, has not actually propelled forward an understanding, because if you deem a particular class of sequence elements to be non-functional, there's no motivation for studying them. And yet, what we have discovered is simply coincidentally, by accident, is that these regions that have been deemed non-functional turn out to be actually critical, in the etiology of many diseases. And so we've really delayed important biomedical understanding because of the commitment that people have to the evolutionary paradigm, which I just don't think is a fruitful way of studying or thinking about genomes. I think, again, a creation model approach, an ID approach is a much more productive format.
Amen. Again, I agree with every single word. We, from the creation or design starting point, we expect treasure. And that's exactly what we're finding. And as you said, we're just in the infancy of understanding the DNA language. So we expect more and more treasure in the future. So again, thank you so much, Dr. Rana. God bless you. Thank you, everybody in the chat. Thank you for all your great comments, feedback, and questions. Standing for Truth is out.